Thank you very much, Jessica. Very interesting. You'll see her a little bit later in the round table with everybody, all the speakers. Now I would like to introduce the founder at the Regenerative Biology at National Cancer Institute. In 2012, focused on the development of tissue engineering methods to address loss of function resulting from traumatic injury and diseases. His research interests are motivated by his military experience. From United Therapeutics, please a round of applause for Dr. Luis M. Alvarez. Thank you.
Thank you very much. Well, it's great to be here in lovely Barcelona. It's my favorite city in Spain. Even though I do have family in León, I don't tell them this. So my name is Luis Alvarez. I recently joined United Therapeutics and established the organ manufacturing group. And I'd like to tell you about what we're doing so that you have an idea of where the field of bioprinting is headed and maybe give you some ideas or inspiration for your own activities. First, I'd like to start with a story of Martine Rothblatt. She's pictured in the center. She originally founded SiriusXM satellite radio. Some of you may actually have this in your cars. Around the time that was going very well, her daughter, all the way on the left, Genesis, developed pulmonary arterial hypertension, which is a usually fatal disease. She decided to leave what she was doing and go into pharma. So she was a satellite engineer who went into pharmaceuticals with the personal mission of curing this deadly disease. And in doing so, she founded United Therapeutics, which brought to market the first drug for pulmonary hypertension. So she licensed the drug from a pharma company, brought it to market, and now Genesis is taking that drug, is alive today because of that drug. So the company originally started around this concept of small molecules to treat pulmonary conditions and has now moved into new areas. So all of the revenues that the company generates for R&D are put into new areas that don't relate to small molecules, but instead relate to regenerative medicine and organ replacement. And this is all driven by the fact that there are, at least in the US, over 100,000 people in various organ types on the organ waiting list, but that number is underrepresented by a factor of at least 10, because if you're too sick to be placed on the list, you're not considered, and if you're not sick enough, then you are not placed on the list. So the true need is actually upwards of 1 million people on the waiting list. So all of the efforts that I'm going to talk about really channel into one idea, which is how is it that we can create one of these. So is it possible now, with the convergence of technologies in 3D bioprinting, in materials, and in mathematical algorithmic modeling, to actually print an object like this? This is a beautiful structure. This is actually a decellularized pig organ, a lung. And if you were to zoom in microscopically, you would see very fine details. And these are very hard to create with today's technology in 3D printing. But that's exactly what our group is trying to do. So last year we announced a deal with 3D Systems that includes the development of a new type of 3D printing platform for ultra-high resolution printing. We also had a deal with 3Scan to generate very detailed organ models. And then in-house, we are developing the inks that are required to print at ultra-high resolution. And I'll talk a little bit about what's in the inks. These are bio-inks that contain human collagen. So how do we start? One of the most challenging aspects of organ printing is this idea that you need to know what it is you're printing. Where do you get the data for an organ? If you were to search the internet, search all published papers, there is no data set for a complete human lung. So we've had to go about doing that. If you look at the lung, the structure includes many different geometries, from the widest branches to the finest alveoli. So there are 480 million alveoli per lung. And you can imagine that that feature is very different than the much larger trachea and larger features. So we have a very challenging data problem. We have a very challenging materials problem when it comes to reproducing an organ. So we can take a medieval approach. You can take a human and chop them up, take pictures, and try to recreate data from that. And in fact, that's what we did. This was not one of our colleagues. This was actually done by the National Institutes of Health and is a data set that you can access as well. So we applied some image processing to extract from this. This looks like maybe a nice steak you might get at a restaurant, but actually there's a lot of data in there. So we applied some of our methods to extract only the lung components. So what is the lung airway in gray? What is the lung vasculature in red or blue? And then to extract the maximum amount of data you can from this data set. So this is actually something that any computer science undergrad can do now. They teach this method. But if you look at this model, it is a static model. It's beautiful, but I cannot make system-wide changes to it in a parametric way. So I can print this object, and in fact we have, but I can't make changes to it. So this has reached a certain limit when it comes to 3D printing of organs and tissues. You can use these methods, but they are probably not going to be sufficient. So now you have to rely on imaging techniques that maybe we're very familiar with, MRI, CT, micro CT, and now nano CT. You can use histology methods. But we've taken another approach that goes even deeper into getting data about a tissue. It's an adaptation of a very old technology, histopathology. If you look at the traditional methods of histopathology, you'll get a very small fraction of the information in a tissue, right? You're slicing slice by slice. Instead, what we have done is we partnered with 3Scan. They've developed a technique called knife edge scanning microscopy. A diamond knife is passed over the organ and sliced in very thin slices, and light is passed through the diamond. So every time you slice, you collect an image of that slice. And we've used this, in fact, to generate now a very massive data set on the lung. So we have 12 of these robots running in San Francisco, and I'll give you an idea of what this scan looks like if we recreate it. This is the raw data. So we take these data and we use that to not only create a structure of what the lung looks like, but to now extract mathematical rules about how to create a lung de novo. So we're creating a lung in silico based on the rules that we get from this data set. So using that, we can actually generate the lung structure using nothing but mathematical equations and rule sets from that data set. So this is an example of the very first mathematically generated lung model, which we're only showing you the first 4 branches, but we're extending this down to all 23 branches. And what this allows you to do is to say, I want to change every vascular lumen diameter by X percentage. And you can do this. In the other models that were static, like I showed you before, this is not possible. So I think to truly represent the tissue in a way that's amenable for 3D printing, it must be a dynamic, parametric, generative model. So for example, we can actually test whether or not these generated models are perfusable. Here we have a bit of a dramatic demonstration, a bleeding printed object in plastic resin, before you go into printing in the more expensive and precious biomaterials. So what technologies do we think are going to enable 3D printing in the future? So the basis of our work is really to think very carefully about that. Are there disciplines that we're not considering now that we need to bring into the conversation so that we can develop a platform that can actually recreate the structures that I'm talking about? So for example, you may have seen a lot of hype. I wouldn't say hype, but it's a first step around this idea of extrusion printing for bioprinting. This is acceptable for research tools, for interesting studies, but I don't think it's going to get us there when it comes to actually solving true human disease, because you need to be able to print a vascular bed, and extrusion methods have a physical limit in that regard. You cannot really recreate vascular beds de novo from extrusion-based methods. So we think that we're going to need other fields, microfluidics, optics, and very novel biomaterials to be able to do this. And in fact, our team has been built around this concept of introducing these new methods. So the methods I will show you now are really optically based methods that are hybrids of the other printing approaches you'll see here in the booths. So SLA, digital light projection, these types of approaches are ones that we've adapted for our purposes. The other thing I'll mention is the biomaterials. So we have also started a brand new project to develop a source of human collagen that is recombinant. And we're able to make this in plants. So in fact, this is a product that's already available in large quantities, and it is human collagen. So if you're going to print an organ, you have the option of using collagen that's on the market. Most of that is from bovine, from cows, or from pigs. And obviously that may present some immunogenicity challenges. So we're starting from the very beginning using human recombinant collagen as a base component in our inks. And in fact, we're making it at very large scale in plants. So actually it has a net negative effect on the environment. And ironically, we're doing this in tobacco. So a plant that was once used to destroy lungs, we're now using it and harnessing its power to make lungs. So I'll show you an example of what the first successful printing of a human trachea scaffold looks like. Here we're printing using an SLA method, right, stereolithography, in an ink that contains human collagen. Of course, many other components that are optimized to allow the features that you want to create. You know, one of the challenges in 3D printing is how do you get a one-to-one fidelity between the object, the digital object that you want to print, and the actual printed object. So that's the essence of high-resolution bioprinting, is controlling those parameters so that you can get objects that actually represent what you need them to print. One interesting aspect of this as well is that this material is fully compatible. So it contains human collagen with other polymer components that degrade at specific times. So that by the time you implant this in a human, those polymers will largely be hydrolyzed away. And I'll talk about what the effect is of placing cells on the scaffold and what cellularization does to these scaffolds. Sometimes things don't go as you planned. So I'll show you an example of what happens when you don't control the reactivity of your inks. In some ways, these inks are living, and they behave as a living object. And this is a funny example that we sometimes call the 3D printed tumor, because of how explosively it pops out of the vat. So this was a failed experiment. We learned a great deal from this. But actually, it was very pleasing to see because we noticed that the reactivity of the ink is well preserved. So one of the interesting things is that once you begin to print objects, here's an example of a human collagen printed trachea scaffold with the properties of a native decellularized scaffold. So for example, elastic modulus between 5 and 100 kilopascals is what you would need to maintain the tissue properties. And anyone who touches this object seems to be fascinated by the lifelike nature of it. It's quite an interesting to see the facial expressions of people who are holding the first printed object of this kind. So what do you do once you make an object? Of course, one of the next steps that's the most important is adding cells. And we have a group in North Carolina that devotes all of their efforts to finding out methods of placing human cells onto scaffolds. They're using pig scaffolds. Eventually, our technology will converge with theirs, and we'll be using our scaffolds to cellularize. So to give you an example of what cellularization looks like, here's a pig lung that is being recellularized with human cells. And if you pay close attention to the histology, you'll notice that the recellularization, the location of nuclei, is very similar to native tissue. In some sense, the cells understand what to do and how to behave because the geometries and the structure of the material is very familiar. So if man can give biology 90% or 80% of the instructions, biology can do the rest. And so our hypothesis is that we can apply the same reasoning to a 3D printed scaffold. So I'd like to conclude the talk just by saying that we're a very open group. We're based in Manchester. We're always hiring, so looking for new talent. In fact, we start very young. This is one of our engineers who brought his daughter by. But we have an active co-op program, and we do bring in engineers from all over the world to work with us for a period of time. If you're interested, please do contact us.